assessment of plant-incorporated protectants. So, Shannon?
All right, thank you. Hello, everyone. I'm Shannon Borges, and as mentioned, I'll be talking to you about the environmental assessment of PIPs. And this slide shows you the 5 different pieces of information that we require to complete our risk assessments for ecological effects. That includes environmental exposure, non-target organism hazard, horizontal gene transfer to soil organisms, gene flow and development of invasiveness, and threatened and endangered species. And I just should note, I'm going to give you a little more details on all of these, but not all of these necessarily require a data submission, and I'll explain more in my next couple of slides. So as you can see here, kind of, is this what's showing on in the webinar? I guess it is.
You know, the individuals can actually change it on their desktop.
Oh, okay.
So.
All right, so folks can see those. Okay.
Right.
All right, so for environmental exposure, we basically asked for 3 different types of information. One is the expression of the PIP pesticidal substance within the plant. Another is the fate and distribution of the PIP plant material in the environment. And also the degradation of the PIP pesticidal substance in the environment. And of these, we typically require a data submission for the expression and also the degradation within the environment. Generally, the fate and distribution of the PIP plant material is something that we determine on our own, but we may ask for some data. And I have some more information here on those. Oops, sorry. Okay, so PIP expression data, these are data that are received as part of the product characterization. They're usually greenhouse or field data. But even though they're part of the product characterization, we do use them to assess exposure, potential exposure to non-target organisms. These are typically expression that's measured in leaves, roots, pollen, grain, whole plant, senescent roots, forage, and stover. And the data are collected typically over the season for certain tissues or organs like leaves, whole plants, and roots. The distribution of the PIP plant material in the environment, this is what determines what non-target organisms may be exposed where and when. It also determines the potential extent of exposure beyond the planted field, and is defined generally for the crop, and it's applicable among different PIPs. So we kind of generally have an understanding of what the plant distribution in the environment will be for corn or cotton or potato or something else. And as I mentioned earlier, we wouldn't necessarily require a data submission for this unless it was something that we didn't have a lot of familiarity with or couldn't find information on. So we may ask for more specific information to be submitted to us for certain crops. For the fate in the environment, we typically look at the soil, the degradation within the soil. And that generally just involves one study to calculate the half-life, or the DT50. And these are typically done as measured in field soil that's brought into the laboratory and created and recreated into microcosms. And typically this also involves molecular analyses that are supplemented with bioassays. And the bioassays are done to provide information on what is actually still bioactive. And it kind of supports the molecular analyses that are done. We have also asked for 3-year soil persistence studies. This was something that we did for BT-based PIPs a long time ago when we were first registering them, when there was a lot more uncertainty. After collecting these data after receiving these data on several active ingredients, we determined that we had enough information to make a determination for those types of PIPs and determined that we didn't need those data anymore. But it is something that if we had something unique come up that we had a high level of uncertainty with, we may ask for those types of data again. So to assess the hazard, we ask for, we have a tiered non-target organism testing scheme, and that is shown here on this next slide. And it basically consists of 4 tiers. The first is high-dose testing, and this is supposed to be something that provides a worst-case scenario. And the second is lower dose testing using the plant material. So that's a laboratory test that's done that's more realistic to what happens in the field. Then there are Tier 3 studies, which are more long-term laboratory studies and semi-field studies, and Tier 4 studies, which are field studies. And the idea behind this testing is that as you progress, as you work through the tiers, you don't actually advance unless you have effects at the lowest tier. So if you do your high-dose testing with your artificial test material and you see no effects, then you don't have to move on to Tier 1. That usually satisfies the testing that we need to determine hazard. If you do observe, if you do have effects at Tier 1, then you move to the next tier and forward as needed. And so the idea is that you're testing worst case scenario first, and then as you progress through the tiers, you test something that's more like what realistically happens in the field. I will say that sometimes there are cases when we have high levels of uncertainty where we may ask for Tier 1 tests and also as follow-up testing, post-registration, we might ask for some field studies. But again, that was something that we required, say, for BT. There was a high lot of experience with those at this point. We haven't been asking for those data, but it is certainly an approach that we could take in another case where we might have a lot of uncertainty. Come on. All right. Oh, and here's just a little bit more information on the Tier 1 and 2. As I had mentioned, the Tier 1 is usually a maximum hazard dose study, so it's, like I said, it's performed with artificial test material, and it's designed to maximize any toxic effects of the test substance on the non-target organism. And it's a worst-case exposure, so exposure is supposed to be several times higher than what's expected under realistic conditions, so 10 times the expected environmental concentration, or EEC. And mortality is usually the endpoint, but sublethal effects may also be observed. As I mentioned, failing Tier 1, you would advance to a higher tier, but that doesn't necessarily mean that there's unacceptable risk. It's just that we need to refine the testing to better define it. And so Tier 2 is lower dose testing with the plant material. And usually the types of endpoints that are observed are the same as in Tier 1. And I will say that in some cases, these studies are kind of limited for certain crops, like for those that produce secondary compounds that might be toxic. And so there are certainly exceptions where we can't actually do Tier 2 testing. So this actually says Tier 1 and 2 studies are often submitted to address non-target hazards. So a lot of times we will get both Tier 1 and Tier 2 studies. And an example is some of the avian feeding studies that are done with grain as opposed to dosing with artificial test material. Here's an example of the type of testing that we do require for most PIPs. You'll see it's quite a list of taxa, and but you'll note that some of these, the wild mammals, freshwater fish and invertebrates, estuary marine animals, non-target plants, those are typically addressed with rationales. Usually in that case, exposure, at least what we've typically had experience with, exposure is usually low and testing is not necessarily needed. We also have some special tests like the monarch butterfly testing that we do require in certain cases. And for combination PIPs, we do require synergism studies as well. And this also lists the Tier 4 tests that we have previously required for certain PIP registrations that we currently have not been asking for because the level of uncertainty that we have from lower tier testing is pretty low. So tiers 3 and 4, they typically encompass definitive hazard level determinations. So where we're looking at in tier 1 and tier 2, we might be defining an LD50 or LC50 that is greater than a certain amount. In these, we're typically trying to determine a definitive one. So an LD50 equals, a certain specific number rather than being higher than a number. And these are, like I said, less common than tier 1. So for horizontal gene transfer to soil organisms, we typically don't ask for data. And then for gene flow and development of invasiveness, this generally involves a paper discussion. And I think I have a little more information on these on the next slide. And I'm just going to briefly touch on gene flow because Chris Wozniak is going to follow my talk here and provide more information. But horizontal gene transfer is something that we've generally understood not to be a problem, but it certainly is on the books as, or not on the books, but it's on our radar as something that remains as a point for consideration. And if you want more details about how we have come to our current understanding, you can see the 2010 BT crops reassessment, which is available at the link on the slide. And for gene flow and development of invasiveness, it's important to state here that gene flow itself does not necessarily constitute an unreasonable risk, but we do still have to perform a risk assessment. And it's usually crop specific unless the PIP involves some unique property or advantage compared to other PIPs. And here I list a couple of points that have to be covered in a discussion that would be submitted. And again, Chris is going to go into these in a little bit more detail, so I won't go over them. But you can see we've made determinations about the risk of gene flow and development of invasiveness for certain crops, and that is also available in that same biopesticide registration action document that's at that link that you see on the bottom of the slide. And last but not least, we also do have to make a determination about the potential risk to threatened and endangered species. And these are not something that we typically require data for. This is a determination that we do on our own. And it's based on the potential exposure to threatened and endangered species and their critical habitats, and determined based on the effects and the environmental hazard data that we get. For BT pips, so far the concern, because lepidopterans and coleopterans have been the target, the only threatened and endangered species that we really focused on are endangered and threatened lepidopterans and coleopterans. Thus far, we've been able to make no effect determinations for them, primarily based on the fact that the PIP is limited to the field in which it's grown, largely, and the threatened and endangered species that are lepidopterans and coleopterans typically tend to have very specific habitat requirements, and so there's no possibility of exposure. So I think this is my last slide. I'm just basically providing you the data requirements that we've developed with our experience with BT pips, but of course there are new types of active ingredients that are coming on the market, one of which is the double-stranded RNA. And so just to give you an idea of what's different for double-stranded RNA-based pips, we do have to make consideration for the unique properties that double-stranded RNAs have compared to BT-derived pips. So these could involve off-target and other unintended effects. There are certainly, we have to make consideration for latent, sublethal, and chronic effects that haven't previously been considered for BT pips. Also for effects on life stages that we haven't typically tested, and the potential variety of molecular structure of these that might affect environmental fate. So these these are things that we're still considering, and the data requirements are to be determined at this point. However, we recently had a science advisory panel meeting for a DVSNF7 double-stranded RNA, and what we required for the risk assessment for that was some additional invertebrate testing to include reproduction endpoints for certain insects, and also a modified feeding study for birds. It was modified to account for a longer observation period to allow us to observe any latent effects that might have occurred. We also didn't require it, but we did receive data on degradation in sediment and water. And like I said, it was not required, but it was very helpful in the risk assessment. And as I mentioned, it was a subject of 2 SAP meetings in January 2014 and most recently in September 2016. And the 2014 SAP did have some influence on what we required for this particular registration, and this latest SAP, it remains to be determined how that will affect our risk assessment at this point. And so I think that's the end of my talk. And with that, I'll hand it off to Chris Wozni.
Thank you, Shannon. Yeah, I'll ask again, if there are any questions or discussion points for the environmental assessment. We can certainly wait a minute, and please feel free to type those in to foster some discussion.
Do you want to sit here?
Yeah. So I think I'll go ahead now and do the gene flow presentation. All right. So you've heard introduction to gene flow that Shannon gave in the previous discussion here. So I'm going to take it a little step further and first give some historical background to how we approach the issue of gene flow and plant-incorporated protectants. And I will not address the horizontal gene transfer issue. I would say Shannon mentioned it, and we typically have recognized that it occurs on evolutionary time, but it is not considered an environmental risk for field testing or even commercial cultivation of pips. So people have generally presented rationale, papers documenting the extremely rare frequency of its occurrence. So with that, on the first slide, I want to make it clear that really what we're talking about here with the gene flow is pollen transfer and hybridization with introgression of a transgene, a PIP transgene, into a sexually compatible wild relative. We're not really talking about crop-to-crop gene flow because, as Mike mentioned this morning, we pretty much require a tolerance action to be in place, or an exemption from the requirement of a tolerance for the PIP trait, such that if your pollen moved, say, to your neighbor's field of the same crop and pollinated it, that's not a violation of FIFRA. As long as there's a tolerance in place, there's no violation with the Food, Drug, and Cosmetic Act. The food is considered safe to eat. So what we're really talking about here is expanding sort of the exposure of the PIP trait to the environment by transfer to a sexually compatible wild relative, typically in a self-sustaining population. So I will say this now, and I'll probably say it 3 more times during my presentation. I don't want to leave the impression that gene flow is somehow a priori a negative consequence, or something to sort of be afraid of, for lack of a better phrase. I mean, we understand gene flow is the basis for the diversity we see in the plant kingdom by and large, and we merely want to get it captured in the risk assessment in instances where the crop does have sexually compatible wild relatives growing sympatrically. And in our case, looking just at pips amongst the broader array of genetically engineered plants, there are very few instances where this is even plausible. I'll talk briefly about what has been done to date in this regard. So in 2001, there was a BT crops reassessment performed, and the document is available online. The environmental section of that covered the gene flow issues for maize, potato, and cotton, which were the three principal PIPs at that time. Now with maize, not too surprisingly, there are no self-sustaining populations of corn in the United States or territories and possessions, so there really was no issue of corn cross-pollinating, say, Zea mexicana or another teosinte or something like that. Those only exist as, museum and special planting kind of things. So that was a non-issue. Potato was slightly more complicated in the sense that there are some wild relatives out west. Most of them tend to either have ploidy differences or endosperm balance number differences, such that there's a genetic inhibition, if you will, for a successful integration of genes. And on top of that, these wild relatives tend to grow at higher elevations. They're not in areas where you'd see commercial potato cultivation, and they're miles and miles apart from any potential cultivated fields of, at that time it was a BT and virus-resistant potato. So again, largely a non-issue. Now with cotton, it was a little bit more complex yet again, in the sense that in Hawaii we have a native cotton, Gossypium tomentosum. Puerto Rico, Virgin Islands, and the very southern tip of Florida all have hybrid swarms of Gossypium barbadense and Gossypium hirsutum, or upland cotton, which are sexually compatible. They're all allotetraploids. And so, we decided we need to take a look at that and just see really what is the potential here for any sort of an adverse effect. Now, I'll point out that mitigation is the preferred alternative when you don't have the data, and that's where we were at in 2001. It has changed somewhat up till now, but we're still to a large degree lacking certain environmental data points. I'll also mention that papaya and European plum have since been assessed and for gene flow issues, and again, non-issues there. Those were much more recent, like 2010-11, when they were, actually the other papaya was just registered last year, University of Florida, which will expand the range, but again, no gene flow issue. So as I mentioned with the feral cotton in South Florida, Puerto Rico, and Hawaii, we had decided that we would basically put in a mitigation measure to preclude hybridization and integration of transgenes until we had further information. At that time, in the early 2000s, we were also fortunate enough to have a little money to fund a couple of pollination biologists and evolutionary biologists to go to Hawaii and actually look at the Hawaiian cotton, because the literature had some information on pollinators. They were arguing that it was crepuscular pollination, meaning that it was pollinated at night, most likely by moths. Turned out that was wrong. In fact, the same pollinators that used to pollinate the feral cotton there would also pollinate the native Hawaiian cotton. There really pretty much is no cotton cultivation anymore in Hawaii, so again, it's turned into a non-issue. Whereas in Puerto Rico, there are winter nurseries for companies to increase seed and do some research on cotton. So we had instituted a 3-mile radius around those plots where there had to be no feral cotton, or they could also institute a trap crop of similar but non-BT cotton surrounding these plots as a means of capturing some of the pollinators coming and going from the fields. And again, I want to reiterate that we didn't look at this and say, it's going to turn cotton into a weedy plant, but we had to know to what degree gene flow is occurring and also what would be the potential impacts to the environment. So, as I mentioned at the outset, one of the considerations is that the exposure to all kinds of organisms in the environment to a PIP transgene product is extended when you go beyond a managed field situation, particularly in instances where you just really don't even know how much has introgressed into wild populations of plants that are closely related to your domesticated crop plant. So again, depending on which species of, crop and its wild relative you're speaking of, there's certainly a myriad of all kinds of insects as well as other plants, other arthropods, and things that will come into contact with that wild relative expressing the transgene. And of course, one of the questions is, what is the impact there, if any, and is it significant? Certainly, there's a potential for a PIP trait to alter a plant habit, the phenology, the timing of the development, and its potential invasiveness. Simply when we, are ignorant of the facts, don't know what's going on, then we have to get some handle on it or provide mitigation measures simply to make the risk assessment complete. Now, when we talked to a scientific advisory panel in October of 2000 about this, we did engage them and ask if a little gene flow is okay, because trying to get to zero is often extremely difficult, if not impossible. And their thoughts were basically that it may change the timing of any such impact that may occur following an introgression event into a wild population. However, it was still gene flow, and there's still going to be some impact if, in fact, the trait was capable of causing such an impact. So one of the things, and I've already mentioned this, but I'll reiterate, there are a variety of invertebrates and other non-target organisms that will be associated both with the crop and its sexually compatible wild relative and hybrids thereof. So that can certainly be influenced by the expression, say, of an insecticidal PIP transgene. And this could include, pollinators, predators, detritivores, all the categories of organisms providing ecosystem services. And that needs to be considered, but I would also mention that a comparative assessment, I think, is good at keeping things in perspective. As we know, gene flow goes on, whether we have PIPs or transgenic plants or not, gene flow is going on all the time. So there is a question there about what's going on in the absence or beyond what's going on with a PIP transgene, for example. In addition, I'll mention, I don't know that it's been brought up today, but FIFRA is a risk-benefit statute as well. And certainly, beneficial sides of PIPs need to also be considered in risk assessments. And these could be things economic, could be fewer tractor passes over a field, so less, carbon dioxide and other pollutants from gasoline engines going in the air. It could be enhancement for farmers, et cetera. In terms of the types of things, we would consider, let's say we had a new species of crop come in that we hadn't seen before as a PIP, and one of the things we'd ask about, well, how is it pollinated? Certainly animal-mediated pollination can be quite different to deal with than wind-mediated pollination. So with some crops, for example, with sunflower, we know that there are bees, various hymenopterans, that will pollinate, and some of them can travel as 5 miles with viable pollen. So the other question is, have people looked at these? Are there models available? Have people actually experimented? All that can be, of course, very helpful and very informative. Wind-mediated pollination, again, a little bit different scenario, but still there are considerations such as crop improvement. Associations tend to have a lot of experience with this. How far do you need to separate the two crop fields to keep pollination at a minimum? I will point out, however, though, that in many cases the distances given are with the assumption that there will be some level of cross-pollination. It might be 0.5%, 1%, 2%, but a lot of these are based on basically what a breeder is willing to live with. Whereas in this scenario where we're talking about trying to preclude the integration of a transgene, until we understand better whether it has a potential adverse effect, we really can't set a level like that and say, well, 1 or 2% is okay, because we simply don't have that knowledge. And of course, there are certain crops that have flowering characteristics, like soybean, for example, that almost preclude, pollen transfer between plants unless they're extremely close. And in other cases, like with potato, in many cases, not all cases, but many cases, a lot of varieties are just sterile, so there won't be any issue there. They're vegetatively propagated. So the consequences of a hybridization event, and really what I mean here is hybridization followed by introgression of the transgene with, penetrance with expression in subsequent generations. Is one of the reasons why, you really need to have a food tolerance in place from the start because, number one, violations thereof where a transgene is expressing in a commodity that does not have a tolerance or tolerance exemption can lead to that commodity be considered adulterated, and the Food and Drug Administration does the enforcement on that side, and they can certainly pull things from shelves. So in that instance, even your neighbor's field is a strong consideration, and that's one of the reasons why we really try to impress upon people when they're doing even small-scale field trials. We typically look at things at 10 acres and above, but even if you're out there at a half acre, and particularly if you're on a university campus where other people are working with the same crop, and I have seen this, where pollen can flow, and it's just a matter of time till somebody finds out that their transgene, ended up in somebody else's variety. And I have seen this also happen with institutions that are basically maintaining germplasm collections, and some of them have gotten contaminated, and it took a lot of money and a lot of time to ferret that all out. In addition, as we know, there are other methods for unapproved presence, adventitious presence of seeds and grain things, and I think we've all seen some horror stories on the trade issues where these have basically ended up in large shipments getting rejected and fines being imposed. So in terms of the consequences as well, one of the things we consider, and I should reiterate if it wasn't said earlier, that PIP, the definition of a PIP product being a pesticide, actually includes some that have potentially plant growth regulatory properties. So you wouldn't expect a BT transgene to change the habit of a plant, for example, but certainly something that altered cytokinin or gibberellin or a ratio of two plant hormones could certainly do that. So one question we would have is, do the seeds still disperse the same between the original wild relative and the hybrid with the PIP transgene? Has there been any change? Does the rachis or the inflorescence shatter and scatter the seed? Dormancy issues, of course, and the ability to volunteer in the following season, particularly if they can form the basis for a self-sustaining population, are all real considerations that need to be taken into account when doing a risk assessment. So weediness or invasiveness, or however you, competitiveness, however you care to characterize the potential, if in fact there is a case where a population is being, say, held in check, for lack of a better term, by a pest of some kind, and now you've provided resistance to that pest to the wild population through gene flow, certainly that could potentially be an issue for the weediness of that sexually compatible wild relative species. Now, I think it's highly unlikely, but can't say the probability is zero, cases where people have theorized about rare alleles in populations being swamped out basically by movement of a selective transgene, whether it's a transgene or even just a normal or standard gene from a crop plant that's been improved, could in theory reduce the biodiversity of allelic distribution within the population. Non-target organism effects, we've already mentioned that. They can be both direct and indirect, larger community effects, and certainly, as Shannon mentioned, there's consideration for endangered or threatened species where there is such gene flow and integration occurring. What makes a plant invasive? That's, I think, the $64,000 question, and I'm not saying I have the answers. We certainly know that there are some characteristics that are worth looking at in depth to see if, again, they've changed or maybe they exist already. Fortunately for us, the overwhelming majority of the crop plants and the ones we consider crops are in fact domesticated in the sense that they tend not to be weedy in their characteristics. There are maybe a couple exceptions. But certainly fecundity, how much seed are you producing and how often, the ability to propagate through non-sexual means, long-lived rhizomes. I'm showing a picture here of purple loosestrife. When I lived up in North Dakota, this was a common ornamental plant that people in restaurants and things would plant it out front, and across the river in Minnesota, it was illegal to sell, and people would probably like to kill you. As you can see, it takes over stream banks and is incredibly competitive and damaging to the local environment. So there are questions about, these long-lived rhizomes that can allow these plants to spread as well as persist through lean times. And again, issues of seed bank dormancy, allelopathy, and we certainly have seen more research in that in the last few years where a lot of plants are capable of producing allelochemicals beyond what we originally had considered likely. In some cases, we may require mesocosm studies if we really had a question about a trait that we thought could impart any of these characteristics to make a plant either overly competitive or whatever. But this is a picture from the EPA lab, the Western Ecology Division lab at Corvallis, Oregon. And these types of experiments certainly are sometimes necessary. You can't do everything in a greenhouse or a laboratory, certainly. So the types of information, if you're coming in with a crop, particularly that we have not seen before, and you've got your PIP trait into it, as I mentioned, this ability to be competitive, is it invasive? Those are the kinds of things that top our list. Is the habit been changed? If we have a natural history of the plant, that's great. Anything in the literature, could be OECD biology of the plant documents, whatever, that gives us some sense of what is known about that plant, again, particularly if it's not one of the typical row crops or fruit crops that we've already seen. Non-target organism effects, again, knowing the action, the intended action of the transgene, as well as the potential for increased exposure by sexually compatible wild relatives in the U.S. Territories and possessions. And of course, community effects, are there other more complex multitrophic interactions that are likely to result from, say, an increase in a wild relative because it's now expressing a selective transgene that gives it an advantage. And as always, the endangered species issues are key as well. And I will point out, I think it was probably already said, but again, impacts on non-target organisms are not totally unexpected. When we're working with pesticides, certainly some impacts are likely and probable, and that's not a deal killer, whereas impacts on endangered or threatened species certainly can be a deal killer. It's a different statute and different standard. So before anyone would start generating information, other than maybe gathering what's in the literature, it's certainly good to just give us a call. I know it was said earlier, I think by all three agencies, that the earlier you call us and discuss things, doesn't cost you much, just a phone call, and we can perhaps be of assistance and planning ahead and helping you avoid some pitfalls. So whether you need to generate some data or not will depend on what is known from the literature, maybe perhaps other research you've already done on that particular trait and trait species combination. I mentioned these OECD documents. A lot of them are very good at covering sort of what's known about the natural history of some plant species. And again, I'll reiterate that a lot of what I'm talking about here is probably more applicable to things other than the traditional row crops that we've already seen, those that are so highly domesticated that, your ability to turn corn into a weedy plant are extremely remote. So certainly any knowledge you have of what the wild populations are, and it's surprising in some cases where you think you could just go to a book or the University of Google and find out what is known about populations. the truth of the matter is, in doing those assessments for cotton and potato in particular, there's a lot of time spent on the phone with experts, plant breeders, evolutionary biologists, ecologists, et cetera, just trying to ferret out, what is really the truth on the ground. So with that, I guess we pause for a minute and see if there are any questions about gene flow or any just comments or points that anyone would like to make. One other thing I will add is that in the past we had done these risk assessments basically for the crop species, and our thinking after that fact was more that we would probably be more reliant on the registrant, on the applicant, producing and providing those materials as far as their thoughts on the potential for gene flow, hybridization, and any potential adverse effects on man or the environment that could result. So not seeing any questions, I think we'll turn it over to the last speaker for today, Alan Reynolds, who's going to talk about resistance management.
Okay, thank you, Chris, and thanks to all the attendees who have hung in there to the last presentation. And I'm going to be talking about EPA's activities with resistance management for plant-incorporated protectants. And I'll preface the presentation by stating that the vast majority of our resistance management experience with PIPs has been with BT crops and insects. But there are aspects of what I'll be talking about here today that I think will be useful for other types of traits and other types of target pests. Okay, so on this first slide, I've got the what, why, when of resistance management. Okay, so first of all, what is resistance management? And simply put, resistance management are use strategies for PIPs to delay the evolution of resistance and prolong the effective lifespan of the registered PIP trait. So why does EPA do resistance management? First of all, there's a risk of resistance that may be greater for PIPs than actually other types of pesticides, and this is because PIPs are typically expressed at high levels in plant tissues throughout the lifespan of the plant. Target pests may have multiple generations per year and may specialize specifically on the BT crop, and these are factors that could lead to higher selection pressure for resistance. Secondly, the PIPs, or at least the ones we've seen to date, have been shown to provide substantial benefits in terms of human health, environment, and economic benefits to growers. These benefits, of course, would be compromised if the PIP or resistance developed to the PIP among its target pests. And then thirdly, resistance to BT PIPs has been considered a reportable unreasonable adverse effect under FIFRA Section 6A2. Okay, so when does EPA require resistance management? Well, certainly we've done it for PIPs targeting insects, and those would mainly be the BT PIPs we've seen to date. EPA may also EPA may also require resistance management for certain target pests that have a history of resistance to pesticides. These could include certain plant pathogens. For other target pests and PIP traits, whether EPA would require resistance management would be determined on a case-by-case basis. We would consider the feasibility of doing resistance management and also risk-benefit considerations. And certainly this is something that we would try to determine very early on in the process. So during the development stage, it's as other presenters have indicated, it'd be very helpful to come in and talk to us early, and we can work through whether we would need a resistance management strategy for the PIP or not. Resistance management strategies have been required to date for commercial use registrations. Those are the FIFRA Section 3 registrations. We have not required them for things like experimental use permits or seed increase registrations, and this is due primarily to the fact that those registrations and permits have lower acreage. The actual resistance management strategy is implemented by the terms of registration for a Section 3 registration.
Yeah.
Okay, we'll get the question at the end, I think so.
Yeah.
Okay. How do I advance the slide?
got to do it. Trigger on the pad here. This mouse doesn't work, and then there's the arrow.
Oh, gotcha. Okay. Thank you. Okay. So on this next slide, I've illustrated the two major components of resistance management. So really two phases. So the first is this risk assessment phase, and this would include data and information that we would use to develop the resistance management strategy. And these would include things like pest biology, the dose of the PIP, mechanism of resistance and potential for cross-resistance, simulation modeling that would be a means to assess and develop a specific mitigation strategy for that PIP and target pest. And then once approval has been granted and the PIP has been registered, there's a stewardship component to resistance management, and these would be the post-registration activities that would actually enact the resistance management strategy. These would include things like resistance monitoring, a remedial action strategy if resistance was to develop, a compliance program that would be needed if there were specific actions required by growers, such as a refuge, a grower education program, as well as reporting to EPA. And I'll be discussing each one of these elements in some more detail on the subsequent slides. Okay, so first looking at the data considerations, we look at pest biology. It's certainly important for understanding the risk of resistance and designing mitigation strategies. So types of things we would look at for pest biology would include life cycle, host utilization, movement behavior, as well as reproductive behaviors. We also look at the dose of the PIP, and what this means in terms of PIPs is pest susceptibility relative to the expression level of the toxin in the PIP crop. So for BT PIPs, we have this concept of a high dose, and this was defined specifically by an SAT panel meeting we had in 1998, and they defined it as 25 times the level of toxin that would be needed to kill a susceptible insect. And that would be such a high level of toxin that essentially only a completely resistant pest could survive. So if you could think of a homozygous resistant insect that is truly resistant, that would be the only thing that would be able to make it through a true high dose exposure. We also look at the potential mechanisms for resistance as well as cross resistance. If we had this high dose scenario that we do for some of the BT PIPs, it's assumed that resistance alleles are recessive and very rare within pest populations. If we don't have a high dose or non-high dose, it could be that there may be multiple resistance alleles involved, or they may be non-recessive or more common in the pest population. So certainly we would like to know what the resistance allele frequency is in the target population. How common are these resistance alleles, which will certainly inform risk. So along those lines, it's very helpful if developers attempt to design selection experiments or create a resistant pest colony. Those can be very helpful to further understand the dynamics of resistance. We also look at the potential for cross-resistance, and this has certainly been a consideration for BT PIPs, where we frequently have more than one BT trait registered for a target pest. So in that case, in those cases, we need to consider, if resistance was to develop to one of those traits, would it also affect other traits that are registered for the same target pests? So for the BT PIPs, we've looked at things like structural comparisons between the toxins, binding properties, as well as common resistance mechanisms that a target pest might have, physiological or behavioral. And all three of these data considerations are very important for the model simulations that I'm going to be getting to on the next slide here. Okay, so simulation modeling is one of the more useful tools that EPA has for resistance management. And they've been primarily used to evaluate the risk of resistance and different resistance management mitigation options that we might have. And these can include things like refuges or IPM strategies, and I'll be talking about both of those in a little more detail in a little bit. So models are specific to the PIP crop toxin and target pests. We've evaluated a number of different modeling approaches over the years, from relatively simple deterministic models to more complex stochastic and probabilistic modeling. These models are parameterized with a lot of the data that I was talking about on the previous slide, including pest biology, dose, fitness, genetics, cross-resistance potential, and landscape features. The output that we typically see with these models is a measure of durability. That could be something like years to resistance or generations to resistance, depending on the target pests. Models that we've seen have been primarily developed by either industry or academia, but now ETA, actually through one of my colleagues, Jeanette Martinez, we now have the capability of conducting our own independent modeling analyses in some cases. Okay, so how does ETA evaluate these simulation models? We specifically focus on really what goes into the model. So what are the parameters and assumptions? Are they realistic? Are they supported by data? We look at worst case scenarios. I think it's one of the more powerful tools we have, and this is particularly helpful when we have uncertainty with some of the biological parameters that we're looking at. We'll look at sensitivity analyses. We want to know, which parameters have the greatest effect on durability. Is that going to be something like fitness or dose, resistance allele frequency, what it is. It's very helpful to get a feel for what's driving the risk. And then, as I mentioned previously, we use these simulation models to evaluate different potential mitigation options. And that's actually a good segue for the next slide. Oops, hang on. There we go. Okay, so what do we mean by mitigation strategies for resistance management? These are primarily would be techniques that would be used to reduce selection pressure for resistance. And they're going to depend largely on the specific pip crop we're dealing with and the target pests. So really to date, we've evaluated two different mitigation approaches. The first one is the use of a refuge, and this has been the primary strategy that we've used for the BT crops. And what is a refuge? So a refuge is essentially non-pip host plants that are available to serve as sources of susceptible insects that would be available to interbreed with any resistant insects that might emerge from the pip crop. And so the amount of refuge, at least for the BT crops, has been linked to the relative risk of resistance. So for pip crops that express a single trait, these are more at risk of resistance, have a higher refuge requirement than a pyramided variety, which would express two or more traits targeting the same target pest. And this concept is roughly illustrated with the graph there on the lower right, showing that pyramids maintain durability with much lower refuge percentage than the sequential use of single traits that are on the right-hand side of that graph. Okay, so in terms of refuge, we've evaluated three different types of refuges, or employed three different types of refuges. The first is what we refer to as a structured refuge, and this is essentially a specific part of the crop that's dedicated to a non-pip variety. And this concept is illustrated in the figure on the lower left of the slide here, in which the refuge is shown as that kind of yellowish-orange fields that are surrounded by green pip fields. And you can see that the refuge is located in an approximate enough way that the susceptible insects or target pests produced by that refuge would be available to interbreed with anything coming out of the pip crop there. We've also had a seed blend approach to refuge, and this is a concept in which both pip and non-pip seed are combined in the same bag, so that makes it much easier for a grower to deploy the refuge. They don't have to manage a refuge separately. It's essentially just right there in the bag, so it just goes through the planter and voila, it's there. We've also had a natural refuge paradigm in which susceptible insects are produced by other hosts. These could be weeds, wild hosts, or other cultivated crops that are also host plants for the target pests. And this has been a strategy that's been used for BT cotton pips in the southeast for the past decade. Okay, so the second major mitigation approach that we've looked at for pips has been the use of integrated pest management. In the concept of, or in the context of resistance management, these would be the use of multiple tactics to manage the target pest and reduce selection pressure on the pip. And really, I think this is the IPM approach differs from refuges in that it's a broader, more holistic approach that's going to likely go over multiple years, whereas a refuge is essentially a year-by-year management strategy. But that's not to say that IPM can't be used in concert with refuge. And that may be particularly helpful where we have low-dose scenarios where there's an increased risk of resistance. An IPM strategy can help buttress that refuge and reduce selection pressure. And I'm going to go through an extensive example with that with corn rootworm in a few slides. Okay, so examples of some of the IPM approaches that might be used include things like crop rotation with a non-host, rotating pit modes of action, if that's possible, if there have been multiple traits developed for that target pest. Use of alternate control measures, those could include insecticides with the pip to sort of create a de facto pyramid, and also the use of scouting to target the use of the pip based on a demonstrated need. And certainly when we think about IPM, there's a large grower component to that, so grower education will be critical to make that work. Certainly EPA, though, will consider other strategies for novel pip traits and new target pests that we haven't previously looked at, provided they address the goal of reducing selection pressure for resistance and preserving the durability of the pip. Okay, so now I'm going to turn to the stewardship aspects of resistance management, the first of which is resistance monitoring. And our resistance monitoring efforts are focused on the major target pests of the pip. And we've looked at two different levels of monitoring. The first is an attempt to monitor changes in susceptibility through targeted sampling and testing, in an attempt to see if there are shifts in susceptibility before they would rise to the level of field-level failure. The second aspect of that is to actually investigate when there is field damage, as that may be a sign of early sign of resistance developing. In terms of a sampling strategy for monitoring, the sampling location should be sufficient to cover the geographic range of the pest, but be focused on areas where the PIP is adopted at the highest levels. These areas presumably have the highest risk of resistance. In terms of detection assays that we would use to confirm whether a population is in fact resistant, there have been several approaches used. First is just essentially a baseline susceptibility measure in which something like an LC50 or an EC50 would be compared with historical averages and also susceptible control populations. A second approach has been to use a diagnostic concentration or a diagnostic assay that's based on a much higher level expression that would approximate an LC99 or even higher. And the goal with this assay approach would be to detect truly resistant insects, those that can actually survive that level of exposure and are presumably going to be resistant to the PIP trait. Primarily, the detection assays we've seen to date have used a diet bioassay, particularly with insects. More recently, we've also seen some on-plant assay approach in which the target pest is exposed directly to the PIP plant. Certainly, there needs to be criteria for defining resistance. For the BT pips, that's been fairly well defined. For a novel pip trait, that's something that we would have to work through. Some of the considerations we've looked at, though, have been heritability. I mean, is this a trait that actually is passed from generation to generation? Is it field relevant? Is this something that we may see in the lab, but what does that mean for actual field exposure? And what's the genetic basis for resistance? Very helpful to know. Okay, now shifting to remedial action, this would be procedures that would be taken if resistance is actually confirmed or is actually determined. And the objective of the remedial action strategy is going to be to contain or slow the spread of resistant populations, particularly to preserve susceptibility in areas where that PIP trait is still effective, or if there are pyramided or stacked varieties, being able to protect the durability of those traits that don't have resistance in them. So there are, to date, at least with the BT pips, there have been two approaches that we've used. There's a level for suspected resistance where that may be defined by damage reports in the field, but we don't yet have an actual bioassay confirmation that definitively determines there's resistance. And then there's confirmed resistance where we do have that actual bioassay confirmation. So some of the strategies that have been used for remedial action, these include IPM-based best management practices, including things like alternate control measures for that immediate growing season or the subsequent growing season, the idea being to try to contain that resistant population. Crop rotation has also been used, as well as other alternate modes of action. A fundamental aspect of resistance or remedial action is to not use the PIP in the area, in the affected area. So I mean, it sounds kind of obvious, but if you have a case of resistance and it's in a defined area, not going in there and continuing to use that trait is one of the key components. Increased resistance monitoring, particularly in the affected area and surrounding areas, is important to understand whether the population is expanding, whether it's moving, or whether the remedial action strategy has been effective. Information, notification to stakeholders is another critical aspect of remedial action. Certainly we want growers to be able to make informed decisions and to take appropriate steps if there is resistance. But there's certainly other stakeholders that need to be part of this process, including crop consultants, seed distributors, university personnel, extension folks, as well as federal and state authorities. And then there are certainly some challenges for, I call them considerations here on the slide, but there are certainly a number of challenges we have for remedial action. First of all, is have these procedures in place before you actually have resistance. It's very difficult to kind of develop a remedial action strategy on the fly. Timing is another aspect, particularly when we're looking at a bioassay confirmation process. How long will that take? In some cases, that may take over a year or more. Defining this remediation zone, is going to be a big challenge for a lot of insects, particularly a lot of mobile target pests. Being able to accurately predict, well, where are you going to focus these remediation measures? Where are you going to draw the line? Presumably, that will be influenced by dispersal biology. But in any case, that'll most definitely be a challenge for certainly any remedial action strategy. Okay. Turning to compliance. Compliance is a component of resistance management when there's a requirement of the grower. The most obvious example of that is the structured refuge requirement. In our experience with BT PIPs, these compliance has been enacted through the use of grower contracts or technology use agreements, and these are between the registrant and their grower customers. For the BT PIPs, for refuge, we have what's called a compliance assurance program, or a CAP. It's a tiered approach that's been actually laid out in very exquisite detail by the terms of registration. The goal of the CAP is really, first of all, it's not to be punitive. We're not trying to punish growers. Really, the primary goal is to bring them back into compliance. However, if growers are still deliberately non-compliant and have repeated incidents of non-compliance, they can lose access to the technology. So it's certainly a disincentive for many growers to not comply with the requirements. As I mentioned on a few of the other slides, our grower education is certainly a very important aspect of resistance management, particularly for the compliance and resistance monitoring components. And again, it's not just the growers that are part of the educational program. It's other stakeholders, including our grower associations, seed dealers, crop consultants, extension folks. Some of the educational tools that have been used have been technology use guides, and these are developed by each company and distributed to their customers that include detailed information about the PIP crop, about the risk of resistance, and what specifically they need to do to lower the risk. Having direct contact with growers is certainly important. It's one thing to get a nice technology use guide brochure, but also having that direct contact with company representatives who can reinforce educational messages certainly very helpful. For BT crops with refuge requirements, we've used bag tags that clearly indicate specifically what the refuge requirement is for that particular product to try to avoid any uncertainty on the part of the grower. Also, electronic media have been used, including things like email, internet, television ads, again, to reinforce educational messages to various stakeholders and growers. And then finally, there's annual reporting to EPA on a number of these stewardship aspects, including resistance monitoring, compliance, and IPM activities. And these reports are very helpful to the agency to be able to assess the effectiveness of the resistance management strategy. Okay. So that, in a nutshell, is the basic overview of what we look at for resistance management. And on the next group of slides, I'm going to turn to a specific case study. And the insect I picked is probably one of our more challenging ones, corn rootworm, but I think it'll be very helpful to illustrate some of the concepts that I've discussed on the previous slide and how we specifically incorporated them into this specific resistance management strategy. Okay, so to date we've had 4 Bt pips that have been registered for corn rootworm, and these were registered starting in 2003 through 2012. Just a few words on the biology of corn rootworm. The name implies the larvae feed on corn roots. They cause damage to that root mass, which can lead to lodging of the plants. That's illustrated in the picture to the right there. The insect has 1 generation per year with an obligate diapause in the egg phase. And both larvae and adults tend not to move a whole lot. Larvae, obviously, they're underground, so it's a little hard to move, but adults as well are not super big dispersers. The refuge strategy that we have in place for corn rootworm includes either a structured refuge or seed blend. For the structured refuge, it's a 20% for single traits and a 5% refuge for pyramids. Similarly, for seed blends, the refuge requirement is a 10% mix for single traits and a 5% mix for pyramids. Despite this refuge strategy, there are certainly some challenges that we have for IRM in terms of rootworm. First of all, this is an insect that has a long history of resistance to conventional pesticides and even some cultural controls that have been used for it. All of the Bt pips that we have registered for corn rootworm, none of them has what we would call a high dose for the insect. So these are all non-high dose expression. In terms of resistance monitoring assays, the diet assays that have been used to date, responses have been fairly variable. We don't have a diagnostic assay that, as I mentioned on one of the previous slides, that could really definitively tell you whether something's resistant or not. So it's been a major challenge to be able to interpret those data to make resistance determinations. Also, just the nature of the insect and its biology with diapause and subterranean feeding makes it a difficult one to work with in a laboratory environment. So perhaps not surprising in light of these challenges, we have seen resistance to some of these Bt pips develop. This was first reported in 2011. And was based on work by an academic researcher at Iowa State University, Aaron Gassman, who went into fields that had damage, collected insects, and he tested them on his own assay system using an on-plant assay, and determined that they were in fact resistant. And since that first paper came out in 2011, there have been a number of other papers that have followed that up and have shown resistance in Iowa, Illinois, and Nebraska, and probably in other places as well. And it was really these reports of resistance that led EPA to reconsider our resistance management approach for corn rootworm and look to buttress that with a better framework. And on this next slide, I'm going to go through specifically what we did to improve the resistance management strategy. So we developed a new framework that has gone into effect. It went into effect starting with the 2016 growing season. It was based on information and guidance that we got from an SAP meeting that we held in 2013, as well as public comment and input from a wide range of stakeholders. The reason we did this is, as I mentioned, we had resistance develop relatively quickly within 10 years of commercialization of these traits. And so our conclusion was that refuge alone has not been sufficient to maintain durability with rootworm. And the resistance monitoring strategies that we have been reactive rather than proactive. So the goals that we had in implementing this framework were to prolong the durability and benefits of Bt traits for corn rootworm. That's consistent with our general resistance management strategy. We also wanted to maintain effectiveness of pyramids. There have been a number of pyramids that have been registered that contain two or more of those 4 traits. And also being able to manage and mitigate cases of resistance where we have them. Because we have resistance in one part of the Corn Belt doesn't mean that these traits will not be effective in other parts of the Corn Belt, and we want to maintain that effectiveness as best we can. Okay, so the main elements of this new framework, and there were 6 of them, and I'm going to go through each one of them in a little more detail. But we have an IPM-based stewardship program that we implemented. We standardized responses to unexpected damage in BT corn fields. We eliminated the requirement to do what we called annual random sampling within the Corn Belt. I'll go into why we did that in a little bit. We use also another one is to encourage the use of on-plant bioassays for resistance determinations. And we also enhanced the existing remedial action plans in the event of resistance. And then finally, annual reports to EPA on some of these new activities, including IPM stewardship and unexpected damage. Okay, so looking first at the IPM stewardship part. So as part of the framework, registrants are now required to develop and encourage growers to follow an IPM-based stewardship plan. The word encourage there is important. This is not a mandate at the grower level. The registrants are required to have a stewardship program and then work with growers, but there's no mandate actually at the grower level. So some of the IPM tools that are part of this stewardship approach include crop rotation to a non-host. That is for a rootworm would be primarily soybean. Also the use of pyramids or alternate modes of action, or just a good old-fashioned non-BT corn. One aspect of it is that EPA sought to limit the use of single toxin varieties, which are more at risk of resistance than the pyramided varieties. So along those lines, each company committed to a phase-out strategy for their single toxin products, such that by 2020, the vast majority of BT corn targeting rootworms should be pyramided. Okay. There's also a recommendation to growers to limit the use of soil-applied insecticides for BT corn, and this is due to some specific resistance concerns that when rootworm exposed to both BT and soil-applied insecticides simultaneously, that could delay their development and create a asynchronous emergence dynamic in which you'd have resistant or BT-resistant insects emerging later than susceptible ones, which could lead to a sort of mating and rapid development of resistance. I've said it before, and I'll say it again, grower education, very important part of certainly any IPM approach or any resistance management approach that engages the growers. The second aspect was to standardize responses to unexpected damage in BT cornfields. This is not really a new concept. This has been on the books for a while. But what we did was essentially strengthen and standardize how registrants are going to handle these cases. And the reason we did this is because with rootworm, these unexpected damage cases are likely to be the real first indicator of potential resistance. And if we're able to execute a timely, effective response, that could help in the overall remediation of that population. So the concept is that unexpected damage exceeding a threshold has to be investigated. And these damage thresholds have been established for both single trait varieties and pyramided products. So if the damage exceeds those thresholds, then corn rootworm will be sampled and then go through this resistance detection bioassay process. But one of the, I think one of the things we really strengthened was before, while the bioassays or this process to determine resistance is ongoing, the registrant will work with the grower to implement immediate mitigation before we even get the results from those bioassays. So they'll work with the grower primarily to recommend that they adopt IPM BMP type approaches to try to manage the population and reduce selection pressure. And these strategies are very similar to what are used for the IPM stewardship program, preferably crop rotation with soybean or another non-host, or using different modes of action. And to illustrate kind of how that works, I've included a little pie chart from, this came from Monsanto, on the right of the slide there, and this shows how, it's a little bit dated, but it shows how growers who had unexpected damage in 2012 responded in 2013. Almost half of them did rotate to soybean, which we think is very effective, but only 8% of those growers chose to plant that same trait the following year. So, over 90% of them, in response to resistance, were adopting some of these DNP approaches that we're advocating. Okay, I'm going to turn to the third aspect. Is we eliminated the annual random sampling, and this was largely because the types of data we're getting were not very meaningful because the same populations were not being tracked over time. And it's extremely unlikely that we'll actually be able to detect shifts in susceptibility before we'll see actual field failure. And this really gets to the non-high dose expression of these PIP traits. And because of that dynamic, it's going to be very difficult to see a meaningful shift in susceptibility that would be below the level of field failure. So rather than waste resources on something that we didn't feel would be a very meaningful approach, we decided to instead more effectively focus on these unexpected damage cases and better detection methods and scouting approaches. Okay, so the fourth aspect was this bioassay confirmation approach. And as I've mentioned before, the diet bioassays have been a bit of a struggle for us, at least from a regulatory perspective, because of the difficulty in interpreting the results and creating actionable levels or actionable triggers in which, if something at a certain level of susceptibility is observed, then something happens. We were never really able to achieve that with the diet bioassays. So instead, what we've advocated is the use of an on-plant approach, which is certainly more of a realistic exposure scenario and can serve more as a diagnostic tool, sort of patterned off the approach that Aaron Gassman used at Iowa State University. That's him there on the lower left. Including, he uses a single on-plant assay approach that can essentially create a very simple but effective determination of whether a population is resistant or not. There's been a second approach that's also been developed called the sublethal seedling assay that was developed by Pioneer that has been used for the Cryo-34-35 toxin. Now, having stated our preference for on-plant assays, EPA is certainly open to other assay approaches, including an improved diet assay approach that can meet the agency's goals of having this diagnostic detection tool. Okay, so the fifth aspect of the framework was enhancing the current remedial action strategies. And these, of course, are the steps that would be taken when we have bioassay confirmation of resistance. The goals, certainly we want to be able to contain resistance, extirpate it if possible, and maintain the durability of PIPs in the areas where they're still effective. Now, the mitigation action area, and as I mentioned before, this is certainly a challenging component to define, but for rootworm, it was defined as 1/2 mile from the damage site, and this was based on corn rootworm dispersal biology. A major aspect of the remedial action strategy is notification to stakeholders. We want to ensure that growers are able to at least make informed decisions and are aware if there is resistance in their county or their immediate locality, that they'll have that information available so that they can make appropriate decisions at planting time. So some of the specific mitigation measures that are used in this mitigation action area. Okay, the first, again, sounds obvious, discontinue the use of single trait products that contain the affected PIP. But for pyramided varieties that have that affected PIP trait and then a second trait in them, if you could think about that, I mean, it's essentially no longer a pyramid. It's really a single trait at that point. So any pyramided product that would contain the affected trait must have a larger refuge. It must have the 20% refuge that we require for single traits rather than the 5% refuge that's used for pyramid. So also in the mitigation action area, continued resistance monitoring, active scouting to assess whether the population is moving, whether it's contained or what. And then also using best management practices to try to control that population, preferably crop rotation, or also use of other BT crops that don't have the affected tip, going back to non-BT corn, or use of adult or larval insecticides, which have the lowest priority in our hierarchy there. So populations can be considered to be mitigated if the grower does rotate to a non-host such as soybean. That's the most effective way we feel to deal with a resistant rootworm population. So if the grower does that in that affected field, we consider that population mitigated. Also, if resistance monitoring shows that there are very few or no rootworm after the implementation of these mitigation strategies, we can consider that a successful mitigation. Or if bioassays show that the population susceptibility is returned to what it was before we saw the resistance event. Okay, so on this last slide, I just included some resources. So there's some general resistance management information, including a URL that has an overview of the agency's resistance management strategies, and that's not just for BT pips, but also for conventionals and also herbicides as well. There's a IRM docket that contains general IRM information. It also contains reviews that the agency has done of resistance monitoring and compliance data and other pertinent information. I've also included one citation here that I worked on with two of my former colleagues that is really a definitive history of resistance management at EPA. So if folks are interested in really understanding in much more detail some of the things I've talked about today, that's certainly a good source. And then specifically for corn rootworm, on the new IRM framework, the URL listed there has most of the details for that. There's also a docket that was established that contains EPA's white paper on corn rootworm, as well as there's a document that goes specifically through the framework, what we did, why we did it, how we did it, those types of things, and also all the public comments that we received as part of the process. So that, Chris, is all I have. So if we have the question.
Yeah, well, we have one question pending that Shannon's going to address on bees. Are there any questions on the IRM section that was just presented? All right, so not hearing or seeing any questions popping up on IRM. Shannon, would you like to address the question and repeat the question? If I recall, it is, does EPA use honeybees as surrogate species for other bees in their risk assessment?
Yeah, I think for pips, I think the answer to that is yes. I know that for conventional pesticides, I think they're looking into potentially testing with non-apis species, but I think there's a lot to be determined there on their side. I can't really comment on that since I don't work on those pesticides. But for pips, and at the moment, the honeybee does represent the honeybee test does provide an estimate of hazard for all other bees at this point.
Okay, well, another second request. Are there any other questions, whether on the IRM talk or Shannon's talk or mine, or matter of fact, we can go pretty much the whole day. Any outstanding issues you'd like to discuss or have us approach? All right, well, not seeing any questions pop up, I certainly like to thank Shonda and Kara and the other folks who helped pulled the technical side of this off, a vast improvement from our last effort, and certainly thank all the speakers as well for their efforts, and as well as the attendees for hanging in there. And I hope that it was informative and something to help you along as you approach the registration process. So with that.
You want to mention the online?
Oh, yes. I will mention I don't have a URL handy, but certainly one of the Biopesticides web pages in the near future, we will have all of these presentations posted for you to download. And I apologize, do not have a URL, but if you were to Google EPA and Biopesticides PIP Data Requirements Symposium, you certainly would get to it. So with that, I think we'll sign off, and thank you again.